Welcome again friends. Welcome to another video tutorial from Shomu's Biology. And in this video lecture, we will be talking about the three different types and modes of RNA processing in eukaryotes. You know, RNA processing is much more complex in eukaryotes compared to in prokaryotes. Because what happens in prokaryotes is that prokaryotic RNA is made in the cytosol, because the genome of prokaryotes also present in the cytosol. And the RNA that is made in prokaryotes, almost in most of the prokaryotes, they carry all the necessary genes that code for proteins. So once they will be translated, they will make proteins. But in eukaryotes, the transcription takes place inside the nucleus. And when the RNA is made from the DNA, that RNA contains two types of units. One is known as introns and second one is known as exons. Exons are the only coding element, while introns don't code for any protein. So what we need to do in eukaryotes, they need to cut those introns out and join the rest of the exons together. That process is known as splicing. After splicing, the mRNA is called matured mRNA. And before not only splicing, but there are two more processes that should be carried out to finally transport the protein mRNA into the cytosol. So before transporting the newly made mRNA to the cytosol in eukaryotes, three major steps have occurred. One is known as Phiprime modifications or Phiprime capping, that is a guanyl residue addition in the Phiprime terminal of that mRNA. 3' polyadenylation, where repetitive adenine residues are added at the 3' section of that newly made mRNA. And then the splicing is carried out, while the introns are cut out, exons are joined together, and that we can use for the mature RNA, which carries the cap, which carries the poly A tail and do not carry any introns. So only exons with a 5' cap and a poly A tail. That is, let me draw it here. This is a 5' cap, then a polyadenylated tail with only exons present in the middle. This is the structure of a mature mRNA, and that mature mRNA is allowed to take into the cytosol for the translation process. Now let's look at all of this process very briefly. I have separate videos on each of those processes that you can watch anytime. Links are provided in the description. Now first is the 5' capping. In the 5' capping, what does that mean? You know, the newly made RNA, they have two different terminals, the 5' and the 3'. In the 5' terminal, they end with 3 phosphate groups, right? 5' phosphate, and the rest of the RNA is played. N means these are the base, the nitrogenous base that are present there. Okay, so between two base there is only one phosphate as a phosphodiester bond over there, and the 3 phosphate is free in the 5' end. So let me draw 5' here. Okay, so we'll look at the process and pathway, but in a very little details. So here you say they involve with the first enzyme known as triphosphatase. The use of this enzyme triphosphatase, what triphosphatase does is it will cleave one phosphate group from the terminal. So phosphatase means the enzyme that cleaves the phosphate group. So now after this cleavage, what we have the structure like this. One phosphate is cleaved. Now after that, we have an enzyme called guanylyl transferase. Guanylyl transferase is an enzyme that only target or take guanine residue and put it and link it at this last site with an inverted link. Normally the guanine is present in the straight conformation, but in this case they put it in the inverted fashion. So there will be guanine and one phosphate group along with guanine, and rest of the phosphates will be present. This is how the RNA will look like. Now in this process they require, remember, GTP, and pyrophosphate will be released because GMP is taken, guanyl monophosphate, and that is added to the 5'. And this is not enough, this is not it. In some eukaryotes, this is the preliminary step. After that, there are further modifications in the 5' end, and that is usually the methylation. I don't know whether you can see the methylation if I draw it here. The methylation is you simply use S-adenosyl methionine. It will transfer the methyl group to the 7th position in the base. So we call it M7 methylation at the 7th carbon in that ring, okay, of this GMP. So there is a methylation done here. Now in some species there is only one methylation event, in some species there are multiple, 2 methylation events. One of the methylation is done in this base in the 2nd prime of the base. The second methylation takes place in the 2nd prime of the base, the first methylation in the 7th in the GMP. Okay, due to this fashion we have 2 different methylation, and in one of the most eukaryotic cells you will see 2 methylations in the 5' capping. This is acting as a cap, and actually this thing helps this RNA to be guided into the cytosol from the nucleus through the nuclear pore complex. That is the kind of advantage of capping. Now if you move to the second modification, that is polyadenylation or poly-A tail, this thing continues along with the synthesis of RNA itself. So let's say this is the double-stranded DNA from where the RNA is being made. So as the RNA is being made, the new strand of RNA, the 5' end is being generated, the 3' end in this direction, right? So at the end of this whole process, there is a specific signal sequence, which is this one. This is the signal, and this is known as the polyadenylation signal. Polyadenylation signal. Polyadenylation signal is nothing but this AAA, this rich sequence repeat that is present here. And there is a specific protein or set of different proteins that involve to interact with that polyadenylation signal at the beginning. When they interact with the polyadenylation signal, they'll bring some more proteins and they'll form RNA cleavage complex or kind of endonuclease activity that can cleave the RNA at a specific point. So there are 2 steps in the polyadenylation. First is this cleavage of the RNA. Second is the addition of adenines in the 3' terminal. So the first step, they need to recognize this sequence, and a protein known as CPSF bind to this region and they recognize the sequence. Then some more proteins known as CSTF, it will also come and bind, and several more protein joins there. I'm not going to talk about the name of the proteins here, but all these proteins are form a RNA cleavage complex. So once the cleavage complex is formed, they will cleave the RNA for a specific point. Now you might ask me, this RNA contains the codons, so if it cleaves the RNA from the middle, how come they not affect this thing in the protein synthesis? The answer is this area of signal, this polyadenylation signal is present at the end of all the codons mostly. So whatever codons that we need are already present here in the rest of the RNAs. This signal presents that this is the end of the transcription process and end of the translation. You don't need it anymore, then only we get the signal. And actually CPSF and CSTF binds there, but they cleave further downstream of it, okay, and they cleave at a specific site, endonuclease activity. So after the cleave, they'll generate the RNA when the specific region or signal region is attached with CPSF and CSTF and many more proteins present as a complex. Now then they will incorporate the protein that actually add all those amino acid, adenine residues there, and that is known as poly-A polymerase. So poly-A polymerase is an RNA polymerase that's keep on adding only one nucleotide, that is A. That's why it's called A polymerase. So it will only bring it's only bring adenine. So it will bring ATP each of the time. In this case it's not actually ATP, they'll only bring the adenine nucleotide that is surrounding, and they start adding the adenine residues one after another, so create the poly-A. Now there are also another protein known as poly-A binding protein or PAB. This poly-A binding protein helps to ultimately find out whether enough number of adenines are attached or not, because they are involved in direct contact with the poly-A binding protein or poly-A polymerase. So you see, this poly-A binding proteins are attached and they try to interact with the poly-A polymerase. So they will ensure that there is enough number of adenine residues attached. Whenever there are enough number of adenine residues are attached, PAB will signal poly-A polymerase that is done, and poly-A polymerase will dissociate and the RNA will be ready. So that is the idea. Now the process of Phiprime capping and polyadenylation may take place excuse me, at the same time. Why? Because the polyadenylation takes place in the Phiprime, which is already free when the start of the transcription if you look, and polyadenylation only required when the 3' gets free, which is further at the end of transcription. So actually the process of Phiprime capping is already done at the early point, then we transfer the adenine residues to the 3' end of the RNA. So this is the first, then the 3' polyadenylation, and once the whole processing is done, then the splicing event will be taken place, and sometimes they might also take place at the same time with this process to make it a little bit faster. Now why we require polyadenylation in the first place? Now cells, especially eukaryotic cells, in those cases this mRNA lifespan are less. Okay, the mRNA lifespans are less because they could be damaged with the exonucleases. There are different exonucleases, they start to cleave from the end of the RNA, and that thing can be blocked. That thing can be blocked if we have several adenine residues attached at the end. So what does that mean? Several amino acids are attached at the end. So even if exonuclease work and act to cleave the amino acids, we still have the intact codon or the gene sequences that we require to be translated. That is the idea of polyadenylation. In some cases it says like it increases the stability of mRNA inside the eukaryotic cell. But again, there are not too much sufficient evidence to support this idea, because there are different examples done. Sometimes we found out that the presence of poly A tail does not matter much in most organisms, but still this is a kind of prescribed manner it might function. Now we talk about the third event, that is splicing. In splicing, there are three different types of splicing depend on the organism that we are talking about. Now as we are talking about eukaryotes, in eukaryotes they rely on a splicing known as spliceosome mediated splicing. It's known as spliceosome mediated splicing. In the spliceosome mediated splicing, what happens? We have two exons. Let's say the normal idea of splicing, what happens is that we need to cleave the introns out, exons to be joined together. So in that case, they always have a specific sequence in the middle known as a branching point. This is known as the branch point. Now the branch point usually consists of an adenine residue. Okay. Now this adenine has this hydroxyl group that is present, the 2' hydroxyl group of the adenine, it is out there. And this 2' hydroxyl, this oxygen have a lone pair of electron that can attack the nearby exon intron junction as a nucleophilic attack. Right? So this type of attack can be done. Okay, so this is the reason of how they form bonds. So let's look at here in this case of spliceosome mediated splicing. Now in there is another splicing called self-splicing, which is more common in the in let's say fungi, in plants which are not that higher order eukaryotes, in protozoas also. They carry the self-splicing. Now self-splicing, we don't need too many complex proteins. It's only the RNA that acts as a ribozyme to catalyze the reaction of splicing. As I mentioned, this hydroxyl can easily attack this joint region, the phosphate bond here, and as a result the bond can be broken down and then they can join these exons together. That is only known as the self-splicing, and this whole process is catalyzed by the RNA itself in those protozoa, in those fungi and lower order eukaryotes. And we take this as an example. There are two different types of self-splicing: group 1 self-splicing, group 2 self-splicing. which is little varied, but more or less the same in mechanism. But in eukaryotes, especially in higher order eukaryotes like us in human, we have spliceosome mediated splicing. In spliceosome mediated splicing, we have involvement of complex ribonucleoproteins. Short ribonucleoproteins come into play. Example of short ribonucleoproteins: U1, U2, U4, U5, U6 and many more. Okay. Now the structure is very similar. Two exons, we have the exon intron terminal here, the middle line is the intron. We need to cleave this intron out, we need to join these two exons together. Now we have a branch point that has A, also have 2' hydroxyl. That hydroxyl lone pair electron attacks this phosphate. And once this attack will be mediated, because in eukaryotic cells, in higher order eukaryotes, two exons are present distantly, more intron contents are present in the middle. So for this A to attack, they need all these locations in close proximity. The only way we can bring them close proximity, if we fold the genes, if we fold the RNA in this case. So we fold this RNA, and for folding the RNA, we need the association of U1, association of U4, U2 and U6. First only U1 comes in, and U1 and U2, and the U2 actually attaches at this place. And U1 comes in and attached to the first exon intron junction. They'll recognize the regions of the splicing, because the first reaction will take place between the branch point and the first exon intron junction. So then they will bring a triplet enzyme complex, that is U4, U5 and U6 together. Once this three comes in, U1 is kicked out, U4 is also released. So U4 and U1 will be released after that. So what we will get here? We will get a structure this is the location of adenine, and we'll have U2 attached with U6, U5 here. So this will be the structure. U4 and U1 will be released. U5 will hold exon 1 and exon 2 together. Okay, as you can see, it's holding together. Now A is very close to the point or the point of cleavage. So as they are very close in the point of cleavage, now it will attack the phosphate, it will cleave it. So as a result of this cleavage, what we will get? Let me draw in this side. We will get, so this part will be cleaved. This is U5, exon 2, it is attached to the E1 to this base part, and then the hydroxyl that is generated in this exon 1, it will attack the phosphate here to the second exon intron junction, and as a result of that, what we will see that exon 1 will be paired with exon 2, and a structure called lariat will be released. So exons will be joined, and the middle part, it will be cleaved out, that is the intron, completely cleaved out, and we call it as a lariat because it looks like a lariat. Okay, so this is the idea. So remember, the first step is to bring this branch point close to the first exon intron junction or terminal. There, once we bring them close with the help of U1, U4, U6, U2 and stuff, U1 and U4 will be released after this close proximity thing is done. Then U2 will be attached to the branch point, so as U6, and U5 is holding exon 1 and exon 2 together, because if we cleave it from there, exon 1 will be released. So before that, now as they bring in very close proximity, the first step of the reaction takes place, which is the attack from A towards the exon 1 intron terminal, so that they'll join here after this cleavage. Okay, and exon 1 is now free, but that's why U5 is required, otherwise exon will be released in somewhere, it will be lost. So U5 is holding them together, and then the second reaction where the hydroxyl will attack, this hydroxyl, the oxygen with lone pair electron, it will attack the phosphate in the second exon intron terminal and then cleave it. As a result, U5 can hold on to both exon 1 and exon 2 attached with each other, so no introns in the middle because introns is cut out there. So thus we get an exon completely out and intron completely out, and that is only exon, so it will form the mature mRNA with 5' capping and 3' polyadenylation that we can bring and take it out to the cytosol in eukaryotic cell, and in eukaryotic cell in the cytosol it will be translated into desired products of proteins. So that in a sense is all the different steps of RNA processing in eukaryotic cell. There are another few steps known as the RNA editing, but I'm not going to talk about RNA editing here. This I think enough about to know about the whole of RNA processing involving 5' capping, 3' polyadenylation and RNA splicing. So thank you for watching. If you like this video, please hit the like button, subscribe to my channel to get more videos like that, and definitely share this video with your friends. Thank you.